Autocyclic proximity recording was developed to detect the proximity of an individual molecular target to one or more other targets. Central to the mechanism are chemically modified DNA probes. Each target has a probe with a specific sequence. A close distance between targets leads to the repeated creation at physiologic temperatures of molecular records of that proximity. To test the proximity between target A and B, their respective probes bind them specifically via antibodies or other means. Recording begins when a polymerase and primers are added to the system. The primers bind the probes and are extended by a polymerase, copying the barcode and the palindrome segment. When the polymerase reaches the synthetic stopper, it stops and detaches from the probe. The newly synthesized DNA strand is called a half record. It is identical in sequence with the shorter side of the hairpin structure and competes to bind their complementary sequence. The intersection of the three strands moves along the hairpin, appearing as a random walk. When the hairpin segment is closed, the half record will extend out with a free palindrome end. If another half record is extended from the nearby probe, they can reach and bind each other by the universal palindrome segments. Polymerases may then bind and extend both segments, using the partnering half record as the template. To complete the cycle, the polymerases extend through the barcodes and the primer binding segments, releasing the full record. This full record molecule contains both sequences of barcode A and barcode B, as the evidence of the specific interaction between target A and B. After each cycle, the DNA probes are back in their initial states. They can therefore undergo repeated cycles of record generation in their pairing. Records are later analyzed for content and the pairings are reconstructed. In many cases, there will be 3 or more targets in close proximity. Each can be labeled with a uniquely barcoded probe. Records can be produced from any pairs within reach. On the other hand, targets not in close proximity are not able to reproduce a full record. Later, target proximity can be determined by analyzing the records. Autocyclic proximity recording allows us to detect the geometry of more complex structures, such as this hexagon consisting of 7 molecular components. Each component is bound to a unique DNA probe. Many records are then produced from each pair of nearby probes. By analyzing the sequences of these full records, we can reveal their proximity relationships and reconstruct the underlying geometry.